So Monday we heard from Sally Leon about fungal pathogens that she studied and some of the history that came before she studied this. Today we have Greg Gauthier,
Gauthier, you got it. You got it.
Who is an associate prof in the Department of Medicine and who studies fungal pathogens that cause disease in humans. That's his topic for the day. Glad to have you here.
Thank you. Well, I want to thank you for inviting me. I also want to thank Sally Leon for suggesting me as the second speaker. Her work was very influential in helping understand one of the genes we found in Blastomyces, which is a homolog of URBS1, which she discovered and really characterized very beautifully. So my talk is Beware the Dirt, Molecular Pathogenesis of Blastomyces, a dimorphic fungal pathogen that's endemic to Wisconsin. And before I begin, I'd like to start acknowledging those who have helped me throughout my career, members who have worked in my lab, colleagues at the University of Wisconsin, Bruce Klein, who I had the opportunity and privilege to train in his lab and still interact and co-author with him. Wonderful mentor, wonderful person, and same with everyone in his lab. Colleagues in Brazil, France, the Broad Institute, and of course all my co-authors. I want to also thank the Department of Medicine as well as the Division of Infectious Diseases for supporting me, and of course my funding sources throughout the years, which has been the NIH, American Lung, University of Wisconsin, and the Infectious Disease Society of America. So what I'd like to try to do in terms of the objectives of this talk is kind of understanding the importance of blastomycosis to human health in Wisconsin, and then delve more into kind of the basic science, which is how dimorphism influences the pathogenesis of blastomyces, the molecular mechanisms that underlie virulence and dimorphism, and how the biology blastomyces influences diagnosis and treatment. So this is kind of a hybrid talk, kind of try to hybridize the clinical as well as the research aspects. And so I'll start off with the clinical case. This is a 38-year-old male without significant medical history, evaluated by his primary care provider for progressive cough, shortness of breath, and fever a few weeks after tubing on the Wisconsin River. His chest x-ray was consistent with pneumonia, and he was prescribed azithromycin. He enjoys spending outdoors and time, including walking his dog. Despite his antibiotics, he became hypoxic, which means he had low oxygen levels. He was admitted to the intensive care unit for medical management. They started broad-spectrum antibiotics, but he didn't get better. He kept getting worse. And this shows his chest x-ray here, and you can see just innumerable tiny, tiny nodules throughout the right and left lung fields. And this is known as a millerate pattern, which is named after a millet seed, which is a tiny, tiny little grain. And unfortunately, he continued to get worse and got whiteout of all 5 lobes of the lung, which is known as acute respiratory distress syndrome. And this was so severe that, that he had to be on a mechanical ventilator, but his lungs were so full of fluid and infection that he couldn't oxygenate through mechanical ventilation. He had to go on something known as ECMO, which is extracorporeal membrane oxygenation, where they take blood out of the veins, put it in a machine to put in oxygen, and then put it back. And so he was admitted here, or actually transferred here from another hospital. They did a bronchoscopy, which means they put a tube down his lungs, squirted some fluid, sucked it back out, and looked at it. And this is what they saw was broad-based budding yeast. So this is a mother cell, daughter cell, and a broad-based bud here between the mother and daughter cells. And so this is very classic for blastomycosis. Question is how did he get his infection? Did he get it from his friends, his dog, that he likes to walk, from the water, or from the soil along the riverbank? And so he got his infection from the soil. Blastomycosis is not transmitted person to person, and it's not transmitted animal to human. And it grows in the dirt, and we'll talk about that a little bit more. So blastomycosis is endemic to the Midwest, South Central, Southeastern United States, along with several provinces of Canada that are highlighted here. So it's geographically restricted in North America. It has also been reported in several countries in Africa, shaded in orange. The overall incidence is about 0.9 or 0.2 to 1.94 cases per 100,000, and there are certain areas in which it is hyperendemic, which means we see a lot of it. This includes Vilas County in northern Wisconsin, up here in Eagle River, which is located in Vilas County, There's the Kenora catchment area in Ontario, kind of around this area, that is also hyperendemic. Blasto is reportable to the Division of Health, and it's been that way since 1994, and that was due to an outbreak in 1984 that led it to be reportable. And over 60% of patients with symptomatic disease require hospitalization. And so this is the 1984 outbreak, which was investigated by Bruce Klein as well as several others. And this occurred, 48 persons developed symptomatic blastomycosis in Eagle River, Wisconsin, visiting a beaver pond. About half were symptomatic. Risk factors for developing blastomycosis was standing on the beaver lodge, picking up objects. Onset of infection was anywhere from about 3 weeks to about 3 months after exposure, so a really long incubation period. And what was really unique about this outbreak is that the Blastomyces was isolated from the soil. And this was the very first outbreak in which Blastomyces was isolated from the soil. And this was significant because the soil samples that grew Blastomyces had a high organic content and an acidic pH, and there was also associated with decomposed wood near the beaver dam. About a year later, there were two more outbreaks, one on the Tamora River and the other on Crystal River. And again, similar incubation period, about 3 weeks to about 3 months. And again, isolated from the soil again, so the second time from an outbreak. And again, this helps solidify the ecological niche of Blastomyces, of where it likes to live in the soil. So it likes forested, sandy soils of an acidic pH that are located near water and contain decaying vegetation. Kurt Reed, who was our clinical microbiology director here in Wisconsin, along with Jennifer Meese and John Archer, who was really of great renown in state epidemiology, did ecological niche modeling of Blastomyces. And they used a number of different variables and mapped it out. And the areas that are in red are habitat that's predicted to be favorable to blastomycosis. And it's a lot of these are along waterways. And you can see there's a higher concentration of red in northern Wisconsin and kind of in the Milwaukee area as well. And when we look at kind of a heat map of human blastomycosis by county of residence, and this was kindly provided to me by Sarah Koski and Susan Gibbons-Berger of the Department of Health here in Wisconsin, shows that there's a lot of cases. Most of the cases, or I should say a chunk of the cases, occur in northern Wisconsin, with more cases being darker blue. But as you can see, cases are reported really throughout the state of Wisconsin. And traditionally, blastomycosis was thought to be due to Blastomyces dermatitidis. However, in 2013, Elizabeth Brown and colleagues were looking at Blastomyces isolates from Canada and Wisconsin, and they actually found that, hey, there's a second species of Blastomyces, and this was named after Dr. Gilchrist. They named it Blastomyces gilchresti after Dr. Gilchrist, who first described blastomycosis in the literature in the late 1890s. He was a professor at Johns Hopkins, and I believe also had an appointment at the University of Maryland. And so, in terms of Blastomyces dermatitidis and gilchresti, there are some differences between the two, especially in terms of ecological distribution. Blastomyces dermatitidis here is found throughout the endemic region. However, gilchresti is a little bit more geographically restricted. It's found in Canada, Minnesota, Wisconsin, and along the St. Lawrence River Valley, kind of in the New York area. And some of these differences are thought to be related to glaciation, because Blastomyces gilchresti is found only in areas that were formerly glaciated, where dermatitidis is found in glaciated and non-glaciated areas, and they were thought to differentiate about or separate about 1.9 million years ago. Well, the story doesn't end in terms of Blastomyces species. Just last year in January, an article came out by Adukic and colleagues finding a third species of Blastomyces that they named Blastomyces percussus, and this was isolated from patients in South Africa as well as one patient in Israel. And what makes Blastomyces percussus unusual is, one, it has a much smaller genome of around 30 megabases compared to Blastomyces gilchresti, which is around 60 to 70 plus MB. Also, the hyphy of percussis is different. There's multiple conidia here on a single stalk of conidiophore. And for Blastomyces dermatitis and Lucrestii, you usually just find one. So there's some differences in morphology. Blastomyces can occur sporadically, which means a single person can get it, but it can also occur in outbreaks. And there's been roughly about 20 well-described outbreaks. Wisconsin has had 10 of those outbreaks. And the most recent one, the Little Wolf River, was 90 patients were infected, and this was the largest outbreak in the state of Wisconsin, and that was 2015. That made the news. And they actually posted health alerts along the river warning people of blastomycosis. And it was associated with tubing, being along the riverbanks, and being on this particular island. At least that's what some of the initial reports came out. There's been a formal systematic analysis of this, but yet to be fully published. So kind of a summary of what we've learned so far, that there are 3 species of blastomycosis, Dermatitis, Goldcrestii, percussis. It's geographically restricted in the United States. It's endemic in Wisconsin, and you acquire it through disruption of soil. You breathe in the aerosolized spores or conidia. Let's do a second case. 54-year-old female with a history of coronary artery disease and high cholesterol developed a progressive cough in late autumn, and she was prescribed levofloxacin. Her chest x-ray here is nice and clear. She did not have any fevers, chills, or night sweats or weight loss, and then in January she developed significant chest and thoracic pain. That prompted a CAT scan, and they found this mass in the lungs here that was not previously seen in late autumn. And there was concern that this could represent malignancy. She has her own business, which is by a river, and she walks to it every day here in Wisconsin. They did a biopsy, and that gave the diagnosis away. So is her clinical presentation most consistent with lung cancer, Cryptococcus, histo, or blasto? Well, since this is a blasto talk, it's blastomycosis. So I want to talk a little bit about kind of how we acquire blastomycosis in a little more detail, and also some of the clinical manifestations. So Blastomyces in the soil grows as a mold that produces spores, and you can see kind of the spores here of Blastomyces dermatitis here. And then soil disrupted, the spores and mold fragments are aerosolized. We breathe them into our lungs, and it converts into pathogenic yeast to cause pneumonia. And about 25 to 40% of patients who develop pneumonia will develop disseminated disease. It can go to the skin, it can go to the bone, and it can go to the brain. And this is a brain abscess here. This is the bone being destroyed. Work out of Marshfield suggests that Blastomyces goldcrestii is more likely to remain in the lungs, whereas dermatitis may be more likely to go out and disseminate. Work for that to confirm that is still ongoing, but that's what a trend has been seen in one of the publications looking at over 300 patients. So pulmonary blastomycosis, this is the most common clinical manifestation. Most people, 60 to 77%, have isolated disease. And the spectrum is widely variable. Sometimes people just have a mild flu-like illness and get better. Some people have subacute pneumonia, which is basically asymptomatic. And that occurs in about half. The other half will develop symptomatic pneumonia, where they get fevers, chills, and feel terrible. And it's similar to bacterial community-acquired pneumonia. You can also get more of a chronic blastomycosis pneumonia that looks exactly like TB, with fevers, chills, night sweats, weight loss. A subset of patients can go on and get cavitary disease, and a subset can even go on and get acute respiratory distress syndrome, which was the first patient we saw. There's no classic chest x-ray finding for blastomycosis. It can do anything. It can look like bacterial pneumonia with consolidation. It can look like cancer with the masses. It can look like a cavity, which you see here. You can get nodules. You can get the miliary pattern, which we've seen before, and you can even get mixed patterns. The most common site of dissemination is the skin, and it can do anything. Verrucous lesions, exophytic, nodule lesions, ulcers, you name it. And one of the classic findings for blastomycosis is having pneumonia with skin lesions. The bone is the second most common site of dissemination, and that results in something known as osteomyelitis, and you get destruction of the bone as shown here, and it can accidentally be misdiagnosed as bacterial osteomyelitis or bone tumor. It can go to the genitourinary system, classically the prostate in men, and symptoms of prostatitis, including painful urination, going often urination at night, urinary retention, suprapubic discomfort. And it's not uncommon for patients with blastomycosis involving the prostate to also have lung and skin involvement. Blastomycosis can rarely go to the larynx, but it's important to recognize because people have been misdiagnosed with laryngeal cancer and actually had their larynx taken out or undergone radiation therapy as part of a misdiagnosis when actually what they needed was antifungal therapy. Usually laryngeal blastomycosis is associated with pulmonary disease. Blasto can go to the central nervous system. Fortunately, it's relatively uncommon in patients without AIDS. However, in someone who has AIDS, it's up to 40%. It can present as a meningitis, as shown here. It can present it as an abscess. It can even have mass lesions. People generally tend to have headaches, confusion. They can have seizures and what we call focal neurologic deficits where they can't move an arm or a leg. Vast majority of people with CNS blastomycosis have widespread or disseminated disease. So to summarize, blastomycosis typically causes pneumonia. It does not respond to antibiotics that are used to treat bacterial pneumonia. That's because it's a fungus. You need to use antifungals. Blastomycosis can disseminate from the lung to nearly every organ in the body, with the skin, bone, prostate, and central nervous system the most common sites. So I'd like to do a third clinical case to kind of now start to delve into some of the basic science. So this is a 45-year-old male without known immunocompromise, developed 28 widespread cutaneous nodules over a period of several weeks. All but 3 lesions on his face and buttocks spontaneously healed. The remaining lesions became progressively larger. He lives in rural Wisconsin, enjoys spending time outdoors, and recent activities include hiking and trimming trees. And these were his lesions on each of the cheeks, so one on the right and one on the left cheek. By the time he presented to medical attention, these lesions had expanded substantially, including pulling down on the eye. This was biopsied. It showed what we call granulomatous inflammation, which is typical for fungal infection, and we saw the broad-based budding yeast, classic for blastomycosis. He was treated with antifungal therapy, and he responded quite well. But with skin lesions with blastomycosis, when you develop them, they tend to scar over. And he did have some damage around the conjunctiva for his eye that did require surgery from our oculoplastic surgeons, and he had a very nice recovery from that. So the big question is what's going on? The last 3 patients we have seen have all been healthy. They're not immunocompromised. They have normal immune systems. For the vast majority of fungal infections that we see in humans, there's an element of immunocompromise, such as having a solid organ transplant, a bone marrow transplant, being on a TNF alpha inhibitor, or very high dose steroids. But for blastomycosis, it's unique. It can infect healthy people. So what features allow blastomyces to cause infection in healthy hosts? Is it thermotolerance, the ability to survive at 37, conversion to yeast morphology, active evasion of the host immune system, or all of the above? And the answer is it does all of the above. If you're a fungus, to cause infection in humans, you have to grow at 37. If you don't grow at 37, you really can't cause disease. And so we have the thermally dimorphic fungi that are pathogenic in humans, and these are characterized by growing as hyphy or mold and environmental temperatures, 22 to 25, and then they're able to convert to pathogenic yeast at 37 degrees Celsius, which is human body temperature. And this conversion is known as the phase transition or the morphologic shift. And in the lab, you can just change the temperature in the incubator and it'll switch back and forth to yeast. It's not a quick conversion. It takes quite a bit of while in the lab to do this. Other dimorphic fungal pathogens include Histoplasma capsulatum, which is here in the Midwest, Coccidioides immitis and Posidacea, which is found in the Southwest and the San Joaquin Valley of California, known as valley fever, Paracoccidioides brasiliensis and Lutzii, which is found in Latin America, especially Brazil, Sporothrix schenkii, which is found worldwide, Talaromyces marneffii, which is found in Southeast Asia, and the Emergomyces species, which are found worldwide. So there are a number of human pathogens, fungal pathogens, that exhibit this thermal dimorphism. They switch morphology based on temperature. However, not all dimorphic fungal pathogens are thermally dimorphic, though there are non-thermal dimorphs. This includes plant pathogens, which you heard about in terms of Oleic from Dr. Liang earlier in the week, but you also have Ophiostoma omi and Nova omi, which are better known as Dutch elm disease, which has wiped out millions of elm trees, Terfrenia deformans, which is a big deal because it causes peach leaf curl and causes about $5 million of agricultural damage per year. Even some insect pathogens exhibit dimorphism. Probably the most famous is Ophiocordyceps unilateralis, which is also known as the zombie ant fungus, and has been featured in National Geographic. And this fungus actually takes control over the ant's behavior. So in terms of our thermally dimorphic fungi, this ability to go from hyphae or these conidia known as spores, the conversion from hyphae or conidia to yeast is absolutely essential for virulence. And this was genetically proven by Dr. Bruce Klein here, who's a professor of pediatric and adult infectious diseases here at the UW. In his lab, they discovered DARK1, which is dimorphism regulating kinase 1. And when you knock this gene out in Blastomyces or silence it in histoplasma, the organism remains locked in the mold phase. It can't convert to yeast. And when you put the DARK1 knockouts or RNAi knockdowns into a mouse, the Blastomyces is absolutely avirulent and the mice survive. So it's absolutely essential. The other thing we find is that in these DARK1 strains, knockout strains or RNAi strains, that they cannot upregulate specific virulence factors such as BAD1, which we'll talk about in a bit. DARK1 is broadly conserved in the dimorphic fungi. It's found in Sporothrix and Paracoccidioides, and it also influences the morphology of Talaromyces marneffii, which is found in Southeast Asia. So in terms of Blastomyces, once we inhale these spores, they can get engulfed by macrophages that live in the lung. However, a portion of them can survive, and it was traditionally thought that Blastomyces was exclusively an extracellular pathogen and had the capability of living inside cells. Alana Sterkel, who trained in Bruce Klein's lab, basically found that yes, Blasto can live in cells, so it has what we call a facultative intracellular life cycle because it can live inside cells and outside cells. And so in these Blastomyces spores, which are in red, are ingested by alveolar macrophages in green, these spores germinate into replicating yeast. And actually, by being in yeast, the transition from conidia to yeast is actually accelerated. It goes faster. And so thus, infected alveolar macrophages can help propagate infection. And this ability to live inside cells is also seen with other dimorphic fungi like Histoplasma, Sporothrix, Paracoccidioides, and Talaromyces. I'd like to talk a little bit about BAD1, which was also discovered by Bruce Klein here at Wisconsin, and it's named for Blastomyces adhesion. It's yeast phase specific, which means it's only expressed in the yeast phase, not in the hyphal or mold phase. And it's an immunodominant protein adhesion. So it's made, it's secreted, some of it binds back to the yeast cell wall, as shown here, and some of it remains soluble. And this multifunctional protein facilitates adhesion to lung tissue and inhibits a critical cytokine for immune defense against fungi called TNF Alpha, and it does it through two different mechanisms, TGF dependent and independent. TGF is tumor growth factor beta, or I should say transforming growth factor beta. And it also affects our CD4 cells, and that can pair the activation of CD4 T cells, and that's important because our T cells basically orchestrate the entire immune response to fungal pathogens, so it impairs their activation. It also decreases cytokines such as IL-17 and interferon gamma, which are also important in controlling fungal infection. So Blastomyces basically modulates the immune system of a healthy host to allow it to establish infection. BAD1 is an absolute essential virulence factor. If you knock it out, and which means you basically eliminate the gene in Blastomyces yeast, and then you inoculate mice with that knockout strain, all the mice survive. It's incapable of causing virulence or infection. So besides BAD1, there's probably other virulence factors. And in a collaborative effort between my lab, Bruce Klein's lab, and the Broad Institute, we did what we call in vivo transcriptional profiling with the goal of uncovering novel genes in Blastomyces that are important for pathogenicity in vivo during pulmonary infection. We used the standard laboratory strain, and what we did is we isolated yeast from mouse lung, purified high-quality RNA from that yeast, and then did deep sequencing, something known as RNA-seq, to look at the transcriptome of Blastomyces. And this has never been done before in the dimorphic fungi. Nobody had ever done in vivo transcriptional profiling to see what's the transcriptional profile of yeast during actual infection. And so...
What were we looking at, sir? What was that photo of?
Oh, this is actually a photo of an organ infected with Blastomyces. This is specifically a lung. And so one of the challenges for this is none of the existing techniques for isolating fungi in mammalian tissue were suitable for what we needed to do. Canada has been done through whole tissue analysis, scraping through oral thrush, enzymatic degradation from kidney infection, and even collecting it from fecal pellets. Cryptococcus has been looked at using a rabbit model of meningitis or through BAL. So we had to develop something new, and as it turns out, I had one of those aha moments in the lab around 9 o'clock where the experiment went wrong, but it actually went right because it led us to a different way of doing the technique. And so what we do is we would harvest the mouse lungs, we'd homogenize them, which basically means almost like put them in a blender, and then we'd spin it down in a tube, and it turns out the density of Blastomyces yeast is different than the density of mammalian tissue, and it's actually denser. So we find the mammalian tissue on top and then the Blastomyces yeast on the bottom, and then we could do a couple quick washes and get really pure yeast, which means we're going to get really pure RNA. And we did some quality control, looking at kind of the RNA bands between mice and yeast and doing real-time PCR to kind of assess, are we seeing what we think we're seeing? And yes, we did have really good, high-quality Blastomyces yeast and very little mouse, very high-quality Blastomyces RNA and very little mouse RNA. And so then we compared our mouse our RNA-seq profile for the RNA we isolated from mouse to in vitro conditions, which included yeast grown in histoplasma macrophage media, mold grown in histoplasma macrophage media, and then we also looked at co-cultivation with bone marrow-derived macrophages in a specific type of media called RPMI. So we did all these comparisons, and this was the bioinformatics was done by the Broad Institute. And when we looked at all of this, we found that 72 genes were upregulated more than twofold. That was independent of temperature, media conditions, or co-cultivation with macrophages. And these were kind of the genes we focused on. And this kind of included secreted proteins, zinc acquisition, transporters, antioxidants, and oxygenases in amino acid metabolism, kind of broad categories. And what I'd like to do is talk a little bit about a few of these. And so one of the interesting things is we found that genes involved in zinc acquisition were upregulated during murine infection in Blastomyces. This includes production of what a zincophore known as PRA1 and Candida albicans, and that's basically made in the cell, secreted out, binds zinc, and brings it back, just like a siderophore that Dr. Leong talked about earlier this week, but it does it for zinc. And we also found upregulation of some high and low affinity zinc transporters. And PRA1 contains all the expected zinc binding domains that we would expect. And in fungi, model fungi such as Candida and Aspergillus, PRA1 and this high affinity zinc transporter are located right next to each other in the gene and transcribed in opposite directions. But for Blastomyces, they're not located next to each other. They are co-regulated, but they're not right next to each other. And in terms of the dimorphic fungi, histoplasma and Paracoccidioides, which are close relatives, don't even have PRA1. Neither does Cryptococcus neoformans, but Eustolagomatus has PRA1 and ZRT1 close to each other on the genome. So we validated our RNA-seq findings by doing quantitative real-time PCR and found that yes, PRA1 and ZRT1 are really, really, really high upregulated compared to other conditions. And in terms of the potential for virulence, this has been suggested in Candida albicans, in which PRA1 is pH and zinc regulated. And when they knock out PRA1 in Candida, it's unable to lyse endothelial cells under zinc-poor conditions. And there's ongoing work looking at PRA1 as well as zinc regulation impacting virulence in Blastomyces. So that work is actively ongoing now in Dr. Bruce Klein's lab. We also found a gene involved in upregulation, that's involved in nickel transport. And why is nickel important? Nickel is a cofactor for urease, which basically converts urea to ammonia, which results in an alkaline pH. And in a closely related fungus, Coccidioides, urease is important for virulence because it promotes survival in the lung and induces tissue damage. So it changes the pH, and that can induce tissue damage. For Cryptococcus neoformans, it promotes survival in vivo and actually promotes invasion of Cryptococcus into the CNS. And Cryptococcus is well known to cause meningitis. So this may be a potentially important gene influencing Blastomyces virulence. And then finally, I'd like to talk a little bit about cysteine biosynthesis and breakdown, because we found a gene that's involved in the biosynthesis of cysteine, which converts serine to L-cysteine. And then cysteine is then broken down by cysteine dioxygenase, which eventually will get converted to sulfite. But sulfite is toxic to the cells, so the cells don't want it. So they excrete it out, and they use the sulfite efflux pump. And what we found is that cysteine synthase, cysteine dioxygenase, and the sulfite efflux pump were all upregulated in Blastomyces. And to me, this is interesting because there's a dermatophyte fungus known as Arthurderma benihai, and dermatophytes basically infect the skin. They don't cause like deeply invasive disease, they just affect the skin, but they are able to break down keratin. And this release of sulfite actually helps break down keratin, which allows these fungi to survive on the skin. And when you knock out either the cysteine dioxygenase or the sulfite efflux pump, the organism has a hard time living on nails and hair, which are keratinized structure. Blastomyces likes to go to the skin. It's the most common site of dissemination. And Blastomyces is unique in terms of that most fungi, or I should say Blastomyces is unique because of its high propensity to disseminate from the lung to the skin. So there's potential that this pathway may contribute. That will have to be explored. So we've talked a lot about kind of going from hyphy to yeast. What about going in the opposite direction, which is yeast to hyphy, when we drop the temperature and allow it to survive in the mold, and where it goes in the mold form or hyphal form? And so it's postulated that growth in the mold form allows for environmental survival, genetic diversity through mating, because Blastomyces does mate, it does have sex, but it only does it in the dirt, and it only does it in the mold form. And then, of course, it's responsible for the transmission to hosts. You don't get infected through breathing in yeast. You get infected through breathing in the spores and the mold fragments. So this is one of the areas that my lab is also interested in, is the conversion in the opposite direction. In using kind of an insertional mutagenesis or a forward genetic approach, we identified a homologue of Eustoligomatus, URBS1, which Sally or Dr. Long discovered in Eustoligomatus. So we have a homologue called SREB, which we named for siderophore repressor in Blastomyces. It's a GATA transcription factor. And what caught our eye about this particular gene is that when you knock it out, the organism fails to complete the conversion to hyphy or mold when you drop the temperature. It kind of starts and then it kind of gets stuck. Whereas the wild type, really nice conversion, you get very nice hyphy here. And this is a time course over a 48-hour period. And just like URBS1, SREB is involved in repressing siderophores, which are iron-gathering molecules. So it represses the genes involved in the biosynthesis of siderophores as well as the transporters. And I do want to say that when we first discovered or identified this gene in Blastomyces, it was the manuscripts and work that Dr. Liang had published that really helped guide the initial research for this particular gene and really helped sort out what was going on. And especially in terms of this, when we had this, the mutant and the knockout, we had it was like, it was pigmented yellow and it changed the color of the media yellow and like what was going on there. And then obviously reading her research, this is involved in siderophore biosynthesis, and we found that yes, we do see overproduction of siderophores when they should be shut off, and that's what this yellow coloration is. The siderophore is binding iron in the media, causing this yellow discoloration of the media as well as the yeast. And when we did gene expression microarray, which we did of this entire time course, comparing wild type to SRAB, which is about 24 microarrays that we used in total, we found that genes involved in siderophore biosynthesis and uptake were upregulated when they actually should be shut off, and so they're higher in blue here, darker blue. And also we found, like many, many fungi, that genes involved in siderophore biosynthesis are clustered in a particular area on the genome. And then we also did what we call ChIP-seq, where we actually looked to see where is this transcription factor binding in vivo during in Blastomyces. And these are some of the peaks for genes involved in siderophore biosynthesis and transport. So we see binding upstream of GATA transcription factor binding sites. And for Dr. Liang, her work showed that yes, for URBS1, that this binds upstream of genes involved with siderophore production that have a GATA site. And so that work was very helpful to us in understanding how this gene regulates iron metabolism in Blastomyces. We delved further into our gene expression microarray analysis, and what struck us as quite interesting, and we saw this through different bioinformatic profiles, is that we had alteration in triacylglycerol biosynthesis and ergosterol biosynthesis, and these are neutral lipids. And what we found is that the transcription of 7 of the 14 triacylglycerol biosynthetic genes were down in the null mutant, and we also found that 12 of the 21 genes involved in ergosterol biosynthesis were also down. And then we did biochemical analysis of the null mutant and the wild type, and we found for the null mutant, there's reduced triacylglycerol and there's reduced ergosterol over the entire time course for the phase transition from 37 to 22 compared to the wild type, which is in black. But there was minimal impact on diacylglycerol, free fatty acids, or phospholipids. And because of this decrease, we said, could there be alteration in lipid droplets? And lipid droplets are organelles in which triacylglycerol is stored, and ergosterol is also stored in this esterified form. And esterified ergosterol is just ergosterol with a fatty acid on it. And when we used a special stain called Bodipi, we found that where the defect in the morphologic shift was, which was just 24 and 48 hours, we really didn't see a lot of lipid droplets, but we see a whole bunch in the wild type. And we quantified this along 10 micrometers along each of the filaments, and wild type would have about 6 to 7, our knockout had 0 to 1, so a really big difference. So we hypothesized that the biosynthesis of neutral lipids, including storage in lipid droplets, is important for proper Hyphy development following a drop in temperature from 37 to 22 degrees Celsius. So the next thing we did is we said, let's just add back in fatty acids. And so we added in saturated fatty acids, which is our 16-0, 18-0, which is your palmitic acid and your stearic acid. And that translated and restored lipid droplet formations, particularly at the 24-hour time point. And then by 48 hours, lipid droplets were gone, indicating that they're being metabolized and used. The other thing we also found is that it accelerated hyphal growth in the null mutants. So we got some correction of the morphologic and lipid defect that we saw. Importantly, the addition of these exogenous lipids did not affect lipid droplet formation in the wild type or affect hyphal development. So this was specifically related to the null mutant that it improved. We looked at unsaturated fatty acids, which would be your oleic acid, and that really didn't do anything, which was a surprise. We expected that oleic acid would do something because in model fungi like Saccharomyces, oleic acid induces robust lipid droplet formation, but not in Blasto. And then we also predicted that maybe palmetto oleic acid would do something. Well, it actually wound up killing all the cells, both the wild type and the null. So this appeared to be specific to saturated fatty acids. We then kind of honed down on genes, candidate genes that were differentially expressed that we wanted to look at. And these include some genes here in the diacyltriacylglycerol conversion and in the ergosterol pathway. We picked ARE because it's involved in the diacylglycerol to triacylglycerol pathway, but it also stuffs on a fatty acid to ergosterol to allow it to be esterified in a lipid droplet. So we decided to overexpress this in the null mutant and see, would we get restored lipid droplets and would we get improved conversion to Hyphy? And in fact, we did. When we overexpressed it anywhere between 50 to 20-fold, there was increased hyphal development and increased lipid droplet formation. We then said, well, let's look at this just in the wild type and target it alone and see if we can have an impact. And we used CRISPR-Cas9 gene editing techniques to do that. And we got 2 mutants, which are shown here. And for CRISPR-Cas9, what it'll do is it'll induce an indel, which is an insertion or a deletion, and then that will result in a premature stop codon, so you don't get your protein. And we found that, yes, there is indeed a defect in the morphologic switch when we dropped it down from 37 to 22 degrees Celsius. However, we didn't see much of a defect in lipid droplets. It was a little disappointing, but what that suggests to me is that neutral-lipid biosynthesis is likely important in hyphal development, and there are probably multiple genes that contribute to lipid droplet formation. ARE just may be a minor contributing factor. So what we are doing is we're looking at some of these other genes involved in the triacylglycerol as well as the ergosterol biosynthetic pathway. We also plan to look to see, could these genes, kind of work together? You need defects in the triacylglycerol and the ergosterol pathway to really truly recapitulate the defects we see in SREB for temperature adaptation. So to summarize some of the basic science research before I go into the very last section, the shift between Hyphy and yeast is temperature dependent. The conversion from Hyphy to yeast is absolutely essential for virulence. DARK1 mediates hyphal to yeast conversion. SREB, which is a homolog of URBS1, mediates yeast to hyphal conversion when you drop temperature, and it may be through regulation of neutral lipid biosynthesis. So finally, last case, 54-year-old female with diabetes underwent a kidney pancreas transplant, was admitted for fevers, drenching night sweats, mild cough without dyspnea. Symptoms began in the late spring, early summer, slowly progressed. This prompted evaluation in early December by her physician. Chest x-ray demonstrated pneumonia, antibiotics were started, didn't improve her symptoms. She had weight loss, nausea, vomiting, a draining neck lesion. She lives on a farm, has a pond in the backyard, house was renovated. Outside her home, she laid mulch, her husband did landscaping. She visited her sister who lives in northern Wisconsin, and the dog is healthy. And so lots of potential soil disruption here. And this is her chest x-ray showing a miliary pattern both on chest x-ray and chest CT. So, and this is a little bit blown up picture to hopefully help see it a little bit better, and you can see all these little tiny nodules here for the miliary pattern. So I do want to make the point that blastomycosis can occur in immunocompromised patients, and we do see it in immunocompromised patients as well as healthy patients. And we did study our solid organ transplant population with a retrospective study, and kind of the highlights from that is the majority had pneumonia. There was a high rate of respiratory failure, high rate of acute respiratory distress. The rate of disseminated disease was fairly similar to historic controls. And then, so only 36%. It wasn't higher, that's in that 25 to 40% range that we've been reported for healthy patients. We did see co-infection with other pathogens, such as viruses, such as CMV or varicella zoster. The mortality was 36%, and however, if you had acute respiratory distress syndrome, it was much higher at 67%. And if patients were properly treated, none of them relapsed. And this was similar to data that was published after our study from Grimm and colleagues looking at Rush, UIC in Iowa, and some from the Transnet database. So in terms of blastomycosis, just a couple things on diagnosis. For us as clinicians, the most important thing is thinking about it. If you don't think about it, you're not going to diagnose it. And then the question is when to think about fungal pneumonia. So we think about it when pneumonia fails to respond to antibiotics. If you have pneumonia and skin lesions, anybody who has ARDS in Wisconsin should be thought about. If you have blastomycosis in a pet, and I'll explain that. You don't get blasto from your dog. But I'll talk about that in the next slide. Travel or living in an endemic area, soil disruption, lots of outdoor activities, and even some things such as using a community compost pile or even a wood-burning stove could potentially put you at risk. And it's not the stove itself, it's the logs that could be rotting and have blastomycosis by it. So why do we care about the dog? And George Sorosi put this the best. Canine blastomycosis is a harbinger of human disease, and blastomycosis is about 10 times more common in dogs than it is humans because dogs have their nose to the ground all the time, whereas humans usually don't. And so we see a similar distribution in terms of the heat map of the epidemiology for canine blastomycosis as we do as human blastomycosis. And what it means is there's a common source of exposure that your dog and you were exposed at the exact same place or time to blastomycosis. So blastomycosis in a pet can be suggestive of human disease because of common source of exposure. In terms of diagnosis, the classic is smear and culture. There are some non-invasive diagnostic tests such as antigen ELISA that came out in the early 2000s, which really revolutionized our treatment. Serologic testing is not very helpful at all. A brand new test came out of maybe about 10 years ago or so called the beta-1,3-glucan test, but this isn't helpful for blastomyces because it has very little beta-1,3-glucan in its cell wall. For histopathology, we've seen that for one of our patients. We get something called pyogranulomas, which we see granulomatous inflammation with neutrophils. There is a newer assay based on BAD1, so bench to bedside research that is being developed, and it has good sensitivity and specificity for diagnosing blastomycosis. So this is still ongoing research for this. In terms of treatment, basically it's a long course of therapy. It's 6 to 12 months for treatment of blastomycosis. And the treatment decision is based on where it is and how severe it is. Finally, the newest antifungal agents that have been developed, the echinocandins, inhibit beta-1,3-glucan synthesis. We can't use them because blasto doesn't have a lot of beta-1,3-glucan in its yeast cell wall. So that's kind of where our biology influences, the biology of the fungus influences treatment decisions. And then finally, the BAD1 null mutant developed by Bruce Klein is actually the first genetically engineered fungal vaccine. If you inject this null mutant, BAD1 null mutant, under the skin of a mouse, it produces 100% sterilizing immunity in the mouse from pulmonary infection. And what's even more is they've identified that its calnexin is the important antigen, and it's protective against other fungi as well, including P. destructans, which is the white nose fungus. There's ongoing research for that as well here at the University of Wisconsin. So kind of some take-home points for blasto. There's 3 species. Its ecological niche is sandy soil, acidic pH, nearby water, decaying vegetation. Pneumonia is the most common clinical presentation. Yeast mycelial dimorphism is essential for pathogenesis. Yeast for immune evasion, mycelial for survival in soil, transmission, genetic diversity. We've developed a new technique to isolate yeast from mouse lung for in vivo transcriptional profiling, which we have undercarried and identified potential new virulence factors. We identified SREB, which regulates morphology in response to a drop in temperature, and this is a homolog of URBS1. Morphologic defect is independent of iron. It influences neutrolipid biosynthesis and lipid droplet formation, and then the addition of exogenous fatty acids can help complement the defect. And our hypothesis that we're working in testing now is that the biosynthesis of neutrolipids, including storage of lipid droplets, is important for proper hyphal development following a drop in temperature. And finally, I'd like to say thank you for everyone for your attention, and again, thank you for the invitation.
Thank you.
Yes?
So just curious about the treatments. It sounds like they're pretty effective, so if you do have an infection, catch it early enough.
Yes.
Chances are you'll recover.
Yeah, usually there's if you catch it early, there's good outcomes. And the treatments are quite effective if initiated early. The outcomes are harder when you have more advanced disease.
What's the target of the drug amphotericin?
So amphotericin, as well as itraconazole, target ergosterol. And what amphotericin does is it binds to ergosterol in the cell wall and creates kind of a ring or a pore that allows electrolytes and other things in the cell to leak out and the cell dies. So it's what we call a fungicidal antibiotic. It actually kills the fungal cell. Itraconazole and related drugs, those actually inhibit the biosynthesis of ergosterol. And if the fungus doesn't have ergosterol, it's kind of hard for it to survive, especially in the yeast form. Those drugs are more, in Blastomyces, are more what we call fungostatic, which means they kind of cripple the organism enough for it to be effective for treatment, but don't outright, but it takes time for it to get killed.
Do you see that antibody approach and the immunization approach to be something of the future?
I think the immunization approach could be important for the future, especially because, we do see, these fungal infections, more severe in our transplant patients and other patients. So if you had somebody who you knew was going to get immunosuppressed, there's always the potential that vaccinating prior to immunosuppression could provide some immunity, which could be very helpful. The other is if it is shown to be helpful against white nose disease for bats, that could make a big difference.
Have you been able to find circulating antibodies? Has that been studied? Somebody told me that the Amish population, which lives more like we did in the last century, when the children are often barefoot and getting into the dirt. They seem to have, some studies have shown that they seem to have quite a bit of resilience with regard to disease. And so perhaps modern people, urban people, aren't being exposed to a lot of immunogens.
Immunogens, yeah.
And so I'm just wondering about that
Yeah.
in terms of, has anybody looked at?
So following an outbreak, Dr. Bruce Klein actually looked at Blastomyces antibody production in humans and found that for at least 2 years there was antibody production. But in terms of how protective that is, that's not entirely known. But there is, at least based on some of the vaccine studies, cross-protection. So there's always the potential that if you got infected and recovered from blastomycosis, you might be protected from histo or coccidiomycosis. But again, that's not been fully tested or fully proven. And then in terms of kind of exposure to kind of the soil and pathogens in the soil that hopefully don't cause you to get disease, there are things such as for asthma, for children who grow up on farms, for example, they're much less likely to have asthma or atopic symptoms because of that early exposure that's thought to do early exposure that kind of primes the immune system, where those who are kind of more in a sterilized environment are more prone to asthma or atopic type symptoms.
No other questions?
Well, I have a question.
Sure.
You may have said this at the beginning, but if not, is there an average age that this seems to be diagnosed in humans?
Yeah, so we see it anywhere from, it can occur in all phases of life. About 90% of the infections occur in adults. About 10% occur in children which would be basically less than 18 years old. And in terms of adults, kind of most of the epidemiologic studies suggest kind of the median or mean age is somewhere kind of in the 40s and 50s, depending on the studies. That's what's kind of generally seen, but it can occur at any age. I mean, We see neonatal blastomycosis has been reported, basically within the first month of life to people in their 90s, so it can occur at any age.
And do you folks theorize about how many people are undiagnosed but that have it?
Yeah, so we suspect, and this is based on outbreak studies conducted here in Wisconsin. Again, this was done by Bruce Klein as well as colleagues in public health here, Wisconsin Public Health Services, is that about 50% of the people exposed to blastomycosis kind of in an outbreak setting will wind up getting clinically symptomatic disease, and 50% will have kind of silent or subclinical disease that never really comes to clinical attention. So it's a thought that about half the people who get exposed will wind up getting symptoms, but that's in an outbreak setting. Well, now, and that's the best data we have. We don't have a good skin antigen or good antibody testing to really look from an epidemiologic standpoint to really characterize it like it's been done for other fungi like histo or coccidiomycosis. And for histo, for example, about 90% of patients who get exposed are asymptomatic. Only 10% go on and get symptomatic disease. For coccidiomycosis, based on some of the work, it's thought about 50 to 67% are asymptomatic, and then about roughly a third to a half will go on and get symptomatic disease. But we don't quite have that for blasto. Welcome.
Yes, thank you very much.
Thank you.